Welcome to today's lecture on cell biology and the fascinating field of stem cell research. In this script, we will delve into various topics related to stem cells, their differentiation and development mechanisms, interactions with the niche microenvironment, techniques for isolation and culture, use in regenerative medicine and therapies, as well as cutting-edge areas such as induced pluripotent stem cells and microRNA regulation. Furthermore, we will discuss the implications of aging on stem cells, their role in cancer, immune responses to transplantation, and ethical considerations in this field. So without further ado, let us begin our journey into the world of stem cells. Stem cells are undifferentiated cells with the remarkable ability to self-renew and differentiate into various specialized cell types. They hold great potential for regenerative medicine, as they can replace damaged or dysfunctional cells in tissues and organs. There are two primary types of stem cells: embryonic stem cells and adult stem cells. Embryonic stem cells are derived from early-stage embryos and possess the highest degree of pluripotency, meaning they can give rise to any cell type of the body. On the other hand, adult stem cells are present in specific tissues and have more limited differentiation capabilities, usually giving rise to cell types within their respective tissue of origin. The process of stem cell differentiation and development is highly regulated and involves various molecular mechanisms. Transcription factors and signaling pathways play crucial roles in directing stem cells towards specific lineages. For example, the Notch signaling pathway controls cell fate decisions, while the WNT signaling pathway regulates self-renewal and differentiation. Additionally, epigenetic modifications, such as DNA methylation and histone modifications, contribute to the establishment and maintenance of cell identity during differentiation. Stem cells reside in specialized microenvironments called niches, which provide signals and cues that regulate their behavior. The niche microenvironment consists of various components, including neighboring cells, extracellular matrix, growth factors, and cytokines. These factors influence stem cell self-renewal, proliferation, and differentiation. For instance, niche-derived signals can promote quiescence or activate stem cell proliferation in response to tissue damage or physiological demands. The isolation and culture of stem cells are critical for their study and therapeutic applications. Various techniques have been developed to isolate and identify specific stem cell populations, including fluorescence-activated cell sorting, FACS, and magnetic activated cell sorting, MACS. Culturing stem cells requires specialized cell culture media, growth factors, and culture conditions to maintain their pluripotency or lineage-specific properties. Stem cell-based therapies hold tremendous potential for treating a wide range of diseases and conditions. These therapies involve the transplantation of stem cells into patients to replenish or repair damaged tissues or organs. For example, hematopoietic stem cell transplantation is used to restore the blood-forming system in patients with certain types of leukemia. Additionally, mesenchymal stem cells have shown promise in tissue regeneration, such as in cartilage repair or the treatment of autoimmune diseases. Regenerative medicine aims to restore lost or damaged tissues and organs using various approaches, including stem cell-based therapies. In addition to stem cells, tissue engineering and biomaterials are utilized to create functional tissues and organs. The combination of stem cells, biomaterials, and bioactive molecules offers exciting possibilities for regenerative medicine, with the potential to revolutionize healthcare in the future. Induced pluripotent stem cells, IPSCs, are generated by reprogramming adult cells, such as skin cells, back into a pluripotent state. This breakthrough technology, pioneered by Shinya Yamanaka, allows the generation of patient-specific pluripotent stem cells without the ethical issues associated with embryonic stem cells. IPSCs can be differentiated into various cell types and hold great promise for disease modeling, drug discovery, and personalized medicine. MicroRNAs, miRNAs, play a crucial role in the regulation of gene expression during stem cell fate determination and differentiation. miRNAs are small non-coding RNA molecules that bind to messenger RNAs, resulting in their degradation or inhibition of translation. By targeting specific mRNAs, miRNAs can direct stem cells towards specific lineages or modulate their self-renewal capabilities. Understanding the intricate regulatory networks involving miRNAs and their target genes is key to deciphering stem cell behavior and differentiation processes. As organisms age, stem cells gradually lose their regenerative and self-renewal capacities. This decline in stem cell function contributes to the development of age-related diseases and impaired tissue repair. Mechanisms of stem cell aging include telomere shortening, DNA damage, epigenetic alterations, and changes in the stem cell niche. Understanding these mechanisms can aid in the development of strategies to rejuvenate aged stem cells and enhance regenerative potential. Cancer stem cells are a small subpopulation of cells within tumors that possess self-renewal and differentiation capabilities, similar to normal stem cells. These cells are thought to drive tumor growth, therapy resistance, and disease recurrence. Targeting cancer stem cells holds promise for improving cancer treatment strategies, as their elimination may prevent tumor relapse and metastasis. Transplanted stem cells can elicit an immune response in the recipient, which may lead to graft rejection or immune-related complications. Strategies to improve immune tolerance and minimize immune rejection include the use of immunosuppressive drugs, genetic engineering of stem cells to evade immune recognition, and the induction of immune tolerance through co-transplantation of immune modulatory cells or tissues. Stem cell research has raised important ethical considerations, particularly regarding the use of embryonic stem cells and the creation of embryos for research purposes. The ethical debate revolves around the moral status of the embryo and the potential for exploitation. Efforts have been made to find alternative sources of pluripotent stem cells, such as IPSCs, to circumvent these ethical concerns while still advancing the field. In conclusion, stem cell research has the potential to revolutionize medicine. It can be used in regenerative medicine, disease modeling, and personalized treatment approaches. Research on stem cell biology and their interaction with the microenvironment is progressing, aiming to improve human health. Ethical considerations in this field require ongoing dialogue. The future holds promising advancements in medical interventions.